Welcome everybody to our cold climate fruit production webinar series. My name is Amaya Tucha and I will be today's webinar moderator. Today we're going to be discussing grapevine decline, a new disease in Midwest, with Dr. Suzanne Slack. Dr. Slack is an assistant professor of fruit production in Iowa State University, and she specializes in perennial fruit crop production and stress management in apples, cherries, grapes, and other fruit crops. She holds a PhD and a master's degree from Michigan State University, and she also teaches at Iowa State University fruit production courses and post-harvest technology courses. We are really excited to have her with us today, and so please join me in welcoming Dr. Suzanne Slack.
Thank you. So, all right, I'm going to share my screen now. So bear with me one second. Everyone see it all right? Great. Okay, so today I'm going to talk about grapevine decline, which a new thing, a new disease. I'll let you decide as we talk more about it too. So this is from a survey that we've been doing in Iowa over the past 3 years. And I just want to talk about some of our findings. Some of my teammates that work in my lab work with us are Brooke and Xiao Chen. So this is a true collaborative effort. So first things first, what is grapevine decline? Grapevine decline refers to a complex of symptoms and issues that can affect grapevines that eventually lead to a gradual decline in their health and productivity. So it's not sudden death, it's not you go out and see a bunch of dead tissue, it's a gradual decline of the vines. The interesting thing about grapevine decline is it's not really a specific disease causal agent, but it's a combination of factors that include pathogens, environmental stress, and cultural practices. Grapevine decline also can manifest in various ways, and it is often involves a decline in vine vigor, reduced yield, and poor grape quality. So for the most part, there's 3 common factors associated with grapevine decline that we see in the Midwest. The first is fungi. So we find a lot of different types of fungi contributing to this grapevine decline. These disease-causing fungal organisms colonize the vascular system of the vine, which leads to reduced nutrient and water transport, and eventually will weaken the vine as those populations of that fungi grow. We also usually have environmental stress. This could be drought, excessive moisture, extreme temperatures, and poor soil conditions, all of which can stress out grapevines and make them more susceptible to diseases and pests in general. But it definitely seems like excessive moisture and extreme temperatures really help make the fungi have an easier time colonizing the vascular system of the grapevines. And last is cultural practices. So this could be improper vineyard management practices, sometimes even by accident that this can happen. This could include inadequate pruning, so too much pruning, not enough pruning, poor soil management, which was nutrient management or organic matter, incorrect fertilization, all of which can contribute to grapevine decline symptoms as a whole. So whenever I'm talking about grapevine decline, this is a perfect picture of the symptoms that we're describing. So you end up with this mature vine, in this case it's about 10 years old, and we end up with blind areas of the vine that we call skips. So for instance, there should have been some buds here that developed into shoots, but they did not, and down here as well. Meanwhile, the cordon is still alive, as evidenced by the shoots that are formed here from these buds, but there's all these blind spots or skips where there should be shoots coming off. So that's the main symptom of the decline that we're looking at. So unlike regular trunk disease, inside these buds or inside this wood, and we're mainly looking for culprits that are in this newer, younger wood than the ones that are in the trunks. So it's a little bit different than traditional trunk diseases. Here's another picture showing these skips. So you end up with these vines that have just a lot of blind spots, a lot of blind wood. One of the potential interactions too is if vines are exposed to quite a bit of pesticide drift, we can also see an increase in these symptoms. So there's just a lot of factors that go into the overall picture of grapevine decline. So if we take one of these vines that are having some skips or having some cordon death, or the cordon's still alive, but there's a bunch of buds, and we look at a cross section, the first thing you'll notice here in this cross section is there is a lot of these dark areas. These are called cankers. So this is caused by a fungi colonizing the wood. And you can see here that they were able to get in probably through pruning wound and colonize further into the cordon. So we see this, we noticed a pattern in 2022, back a couple of years ago now, and since then we've been doing even more surveying to kind of figure out what fungi are involved. One of the other symptoms that we see a lot is we see low fruit count and delays in ripening, or increased ripening. So in this picture here, this is a 12-year-old grapevine at Verasion, and it had like 4 clusters on the whole vine, which it shouldn't have had. So it was very weakly producing. And in that whole vineyard, so that vineyard had, that block had 36 total vines and about 30 pounds of grapes. So that's completely unacceptable for trying to make a profit or make wines. So this disease can be pretty problematic. In Minnesota and Wisconsin, a couple of years back, they did a large survey of different fungal culprits potentially causing trunk disease symptoms. And what they found were a bunch of different fungi causing problems in trunks. So again, trunk disease versus grapevine decline. Grapevine decline is more of a problem with younger tissue in the cordons. So this was more of traditional true trunk disease work. And they found quite a bit of cytospora, esca, diaporthy, botrosphaerias, alternaria, and then they found some fusariums as well. So it was kind of the usual suspects, so to speak, that they found in the survey, and we were kind of expecting to find the same thing. And the reason why it's important to do this survey work is because different fungicides will control these different pathogens better or worse. So if we can get an idea of a specific vineyard's makeup of what fungi they're having in their vines, we can potentially give better recommendations for both treatments as well as maybe some pruning recommendations. So that's one of the reasons why we wanted to do this survey in Iowa. So what's happening in Iowa? For the last 3 summers, we visited multiple declining vines from vineyards across the state, and we found lots of cane wood and young wood that looked like this. So all of that discoloration that we're seeing is from fungi colonizing those shoots, colonizing cordons. This is a collaboration with the plant pathology department here at Iowa State University to figure out some of these pathogens. So we've been doing this for 3 years now. This will be the 4th year as we visit even more sites as folks are becoming more interested in what they have. So what we do is we go out to vineyards and we collect symptomatic and non-symptomatic wood. Typically, we don't cut into the cordons themselves unless there's those skips that I was talking about. We mostly stay on as younger wood or freshly going wood or wood near those skips. We don't really like taking cordons out from folks. And then we plate them. And what happens whenever we surface sterilize them, and if you put them on a special type of media, the fungi will just come out. The ones that are inside the wood come out, and we're able to see what they are. The plates look a little messy, so we end up having to clean them up until we get single colonies. And then once we get single colonies, we're able to use PCR and use molecular techniques to positively identify them. So one of the caveats or issues with this method is it's slow. It can take up to 2 months, sometimes longer, for this process, depending on how fast the fungi grow. A lot of our wood-dwelling fungi don't grow super fast. So sometimes this can take quite a bit of time to get the results, which can be problematic. So looking at the fungi that we found in Iowa, these are some of the counties we surveyed. This is from July 2024. We've surveyed a couple of other counties since then, but this still kind of gives the gist of what we found from these spur samples that we've been taking. We found quite a bit of alternaria and cytospora, which are things that we expected, as well as a little bit of diaporthy, a little bit of Didymmella, but we found Fusarium and Pestalotiopsis pretty much everywhere from these 30 vineyards. And when we looked at all of the isolates we collected, every single vine that was symptomatic had Fusarium in it, which was unusual. And this is also old. We've actually doubled the amount of isolates since then, but the percentages are about the same. And we found that we found a bunch of different type of Fusarium, Alternaria, and Pestalotiopsis for the most part in these vines. So that was pretty surprising. We didn't see as much Cytospora as the surveys up north. We only saw a couple of Botrysphaeria, which are these Dothiomycetes, or the traditional bot canker. And honestly, we expected to find a lot of bot canker, and we barely found any. So we really focused in on what was going on with this Fusarium and why we were finding so much of it in these vines that had all these skips and all these grapevine decline symptoms. So the first thing that we want to do is make sure that the fungi that we're pulling out of the wood is actually causing disease. Sometimes if there's dead tissue, especially dead wood, there's a lot of different fungi that like to eat that dead wood, and we can pull them out on accident, even though they're not the fungi that caused it. So we create wounds in branches, or create wounds in shoots, and do some infection assays. So this one up top is a control. So we just make the wound, and you can see there's no fungi. It was still quite happy. And this one below would be an extreme case of that fungi causing disease, able to spread really far throughout that grapevine tissue. So we found a lot of Alternaria and Fusarium, so we did some preliminary tests. And in this figure here, this A is an example of the Fusarium infected grapevine, whereas B is the Alternaria infected grapevine as a comparison. So even though some Alternaria could cause disease, for the most part, if they did, it was very, very mild, whereas the Fusarium just knocked the grapevines for a loop. It was pretty pathogenic, extremely pathogenic on especially younger shoots on grapes. So we decided to continue working with it to see what else was going on with these different Fusaria. So one thing to note is we also took different age material. We would take spurs, like I said. We wanted to see these different Fusarium species and kind of where we found them. So on 1-year-old spurs, so that would be current growth spurs, some of these fungi, especially like Fusarium graminearum, which is the one we found the most, typically preferred younger aged materials. So as we get into the older spurs, or we get further back into the spurs as we're taking samples, we find actually less and less Fusaria. So 4-year-old spurs only found a couple of incidents of actually having Fusaria colonizing that wood, whereas the current year's growth or the past year's growth seem to have the most of that. That kind of tells us that this Fusaria is probably just killing the buds and killing actively growing tissue. So it's something that could be potentially controlled through pruning and some fungicide methods, as if we're spraying the right type of fungicides. So that's actually good news. It's not really causing, it's not really moving that far into the wood. It's not getting really into the cordons. It's not really getting into the trunk. It's just affecting those buds, which is why we see those skips. And this is just for funsies. Some of you may have heard of Fusarium graminearum before. Fusarium graminearum is a fungi that's more associated with small grains. So it's pretty problematic on corn, it's problematic on wheat, it's problematic on barley. And whenever we look at our isolates from grapes, however, they actually are they're related, but not as closely related as the ones from wheat and corn. So we can't say for certain that they're coming off the wheat and the corn, but they're closely related, but they're not the same types of strains. So that's interesting to see too. So we can't go, can't blame corn, can't blame the wheat grower or anything like that. So they're kind of like a new type of strain of this specific type of fungi. And we're still working on characterizing them further. They're very pretty. They're pink fungi that's causing most of this problem, which I guess is apt for grapes. But yes, so even though this is a graminearum species, it's not necessarily known if it can jump from wheat to corn, grapes to wheat, grapes to corn. So that's something that our lab's still working on. However, we're not super concerned about it from where your grapes are, where your corn, things like that. So we also decided to do some more in-depth sampling to kind of figure out what this means. So if you have fusarium, is it really that big of a deal? Is it causing you an economic loss? Is it something that you could prune out? So we were fortunate enough to have this 48 panel vineyard that we were able to do some in-depth sampling at. It was Marquette, planted in 2009. In this specific one, these 48 panels, they had about a 46% crop loss. The estimated yield should have been about 500 pounds, and it came in at a little over 250 in 2024. So what we did was during pruning, we went out when the grower was pruning and we sampled 15 panels during that time, so about 33% of the vines. And we found that 60% had fusarium graminearum and 40% did not. So this allowed us to kind of see like how much the vines were losing yield if they had fusarium, since not all of them tested positive for it. So this is the harvest weights in that declining Marquette vineyard. So this is pounds of grape clusters per vine. So if there was no fusarium found, it was about 2 pounds more per vine than if there was fusarium found. So there was a yield loss, about 2 pounds. However, 6 pounds per vine, maybe these aren't the yields that we're looking for. There's probably some other factors going on in this declining Marquette vineyard. Remember, grapevine declines, not just the fungi. There's some other things going on as well. So, but the fusarium did decrease the yield. So the next question that we had was, are there any protectants that could spray at pruning that might increase the potential to get ahead of this disease, prevent different fungi from colonizing those pruning wounds? That's right now is the hypothesis and theory of how some of those fungi are getting in. So for this, we went to a different La Crescent vineyard. This was a La Crescent vineyard that we found fusarium in treatments, and we tried a spur renewal pruning system on these single high wire grapes. We tried paint, resin. We also tried some different products. So we tried Lifeguard, Botcher Stop. We also had an untreated control that we didn't do anything to. We also tried Root Shield, Regalia, and then a Trichoderma. And then we have a comparison of a control. So this was an untreated control that vines had no fusarium found in it. And then this axis is our average grape cluster weight per vine, again, in pounds. And what we found was that the Lifeguard, Botcher Stop, untreated control, and the Root Shield were about the same for the amount of cluster weights. Regalia and Trichoderma did a little bit better, but there was still a loss of harvest weights compared to those vines that had no fusarium found for this spur renewal pruning style. We also had a question that if you did a cane renewal pruning style, would there be any differences? And we did find some differences with that cane renewal pruning. In this case, the botcher stop treatment did just as well as vines that did not have any fusarium at all. So that was exciting to see. The paint did reduce it a little bit, probably from physically blocking that entry wound, but it was a bit of work to put the paint on all those wounds. So we're going to try it again this year. We're about to prune here actually next week, so we'll have 2 years of data on this. But you can see some of the treatments didn't do as well of a job of keeping those wounds clean, and there was still some loss of cluster weights on those vines with our treatments. So here they are next to each other. So in blue is that spur renewal pruning, and orange is that cane renewal pruning. So we can see that for some treatments, like for instance with this botcher stop, the botcher stop in combination with the cane renewal pruning on grapevines that have tested positive for fusarium did increase the yield from about 13 to 16 pounds, whereas some of these other treatments was the opposite. The spur renewal still had higher yields, and there's probably no significant difference between this. It's like 16 and a half versus 16 pounds on our vines with no fusarium found in this lucrescent vineyard. But as you can see, the untreated control, you're going from about 9 pounds on average from 16 pounds. That's a pretty big difference if the vines are infected with fusaria in this case, in this lucrescent orchard or lucrescent vineyard. So there's definitely differences in cultivar. We're finding that Marquette seems to be the most affected by this as far as a yield loss of all our survey. Of course, we grow a lot of Marquette, so there's probably also some bias there from that sampling. But it seems like Marquette's having a struggling time with this fusaria infection, whereas lucrescent's doing a little bit better. So there's some other good news. We did some fungicide screening of different rates of commercial products, and we did find that all of our fungi, all of our fusarium species complexes did, besides oxysporum, but that's more of a problem for trunk diseases. But this incarnatum esquisitae group is where all of the ones that seem to be causing problems are, as well as the Sandbuchium. And what this graph is showing us is that as you increase the rate of fludioxonil in this case, that these fungi are susceptible to these fungicides, and we're currently doing more traditional fungicide screening. So there is some hope that maybe we can control this a little bit better if we're spraying the right fungicides at the right time too. So it's not all total doom and gloom. It's just a new thing that we might have to modify our spray schedules for a little bit in the spring. So my conclusions are grapevine decline is a multitude of factors. It's not just the fungi. The vines have to be weakened or have gone through some kind of traumatic event, whether that be a derecho, a freeze, or like a flood, something like that, as well as usually pruning, too much pruning, not enough pruning, pruning at the wrong time, things like that. At least in grapevine decline in Iowa, fusarium is a major factor. And we found that fusarium kills buds and doesn't really infect deeper into older wood. It kind of stops year 2, doesn't really permeate further than that. So it is something that we can clean up. It's just a problematic that it's killing those buds that are going to produce our fruit. So we see a lot of yield loss with it. But it is susceptible to fungicides. There's lots of different management options that we have to control it from entering those pruning wounds. And we are currently working on spray recommendations and hope to have some of that available this time next year. So with that, I would like to thank my lab group. So these are some of the students and other professors who work with me at Iowa State. We do a lot of work, not just on grapes, but on apples, strawberries, raspberries, pears. If you can grow it in Iowa and it's a fruit, we probably have a project on it. And then I want to specifically thank Dr. Xiao Chenyuan and Dr. Randall Voss for their help with this project, as well as Brooke, Grace, and Emma, who are some of our students who do a lot of work on this specific pathogen system and grapevine decline in general. And with that, I can take questions.
All right. Well, thank you so much, Suzanne. That was really interesting. There's always so much mystery around these trunk diseases. So that's great. Really great new information. There's a couple of questions on the chat. I'll read them both because I think they're sort of related, so probably going to have the same answer. So the first one says, did you guys notice any issues with native wild vines in the Midwest? And then a question about whether there's some specific difference among, I guess, cultivars, which you said something that Marquette was a little bit more sensitive. And then the following question, which might also be related, is the fusarium biotypes that appear in grapevines are distinct from those in corn and wheat. What is the source of the grapevine fusarium? Nearby wild grapes, perhaps?
Yeah, that's a great question. So we are screening wild grapevines. We are screening mainly Riparia and some Labrusca species of grapevines. We're actually finding completely different profiles in those grapes. So we are seeing little bits of fusarium, but they actually have more of our traditional grapevine pathogens. So we find a lot of phoma, we find a lot of Botryopsera and Cytospora in those. We want to increase our search this year. If you have any wild grapevines and you want to share them with us, it's kind of awkward to go up to someone's fence post and just take cuttings sometimes. We usually try to get permission. But we are looking at those. And like I said, for the Riparia, it's more of what we were expecting. The Labrusca is a little bit more all over the place. But I will say, looking at our cultivars, definitely Marquette is really sensitive. And we see it, but again, we have a lot of Marquette. So it's also potentially some sampling bias from that. But one of the things that my students currently looking at is looking at all, I think we have 35 cultivars now, and she's currently running analysis. I wish I had it in time for this seminar, but you know how that goes. That we're trying to figure out if there's any correlation between different pathotypes too, because some of them are more pathogenic than others.
Perfect. We have another question in the chat. Says, how do you know the decline in yield is from fusarium and not bull cane selections in pruning or poor hardening off or bad winter kill? Or pruning woods blocking sap flow?
Yeah, that's a great question. So all we can say is that if you're in a vineyard and they've all been treated the same, so we're assuming, you know, that all the pruning's done the same. Most of these blocks are smaller. We're just looking at if the fusarium's there or not. So we're trying to take out all those other factors that you mentioned, which can contribute to lower yield. Completely correct. But we're mainly looking at vines that have tested positive for fusarium versus ones that have not.
Perfect. I see that Josie has her hand raised. Maybe she wants to ask her question live instead of putting that in the chat. Go ahead, Josie.
Apologies, I must have accidentally hit that. My bad.
It's okay. I have a question though. Suzanne, how can you distinguish between where you think is some of this decline due to the fusarium with the traditional, trunk diseases?
Yeah, that's a really good question. It's just mainly surveying who's there and what's there. So, like I said, we just haven't found a lot of the traditional fusarium or traditional grapevine trunk disease pathogens, at least in the cordons and in those buds that we're sampling. Now, if we did whole trunk, if we took trunks out, we may find some of those pathogens. However, we're mainly interested in vines that are showing those skips and having those lower yields that I was talking about. So we would love to cut up people's grapevines, but you'd be shocked at how many people don't want us to take their whole grapevine back with them. And most of the time, folks will let us take spurs back, no problem. So.
If anybody else have more questions, just feel free to post them in the chat. Oh, here we go. For Marquette, how are they trained and how many buds left? I don't understand the full question, but after pruning, I guess, how many buds are living on the plant?
I'm not going to be able to answer how many buds were left on the plant because it's going to vary from how the grower is managing, but almost, I think every single vineyard that we went to either had single high wire or a double curtain style. but it was definitely more of single high wire. We only have a couple of folks doing double curtain these days. So I will say that on the double curtain places we went to, it was definitely worse. But I am not sure how many buds they were leaving on those vines. I know for the ones that we have on campus that are having disease problems, we do have that data because we do take pruning weights and we do our calculations. But for the grower survey, I can't tell you that.
I also noticed from your data that when you show like the traditional spur pruning versus the cane pruning, that there were some substantial differences in some of the results. Do you think that that's due to, with the fungicides that you tried, do you think that's due to the amount of like cuttings and possible entrance for the fusarium?
Definitely. So like with the cane style pruning that we did, that the grower was doing in that style was a modified Athena. So that's only two to four cuts a year that they're doing on that grapevine versus, potentially like, 30 or 40 with spur pruning. So I think it's way easier to cover those as well. So like you could do paint, you could do resin, you could easily treat, 3 or 4 wounds, but trying to do everything. And then, time of year, do you have your air blaster set up? Like there's a lot of other questions too on how you would manage covering all those wounds on a, big scale vineyard. It was really difficult for us to spray those wounds for our spur pruning. We did our best and we were trying actively to make sure we hit every single one. And I think that might be hard.
And so in the case of trying to protect the vines, like can you explain us how fusarium would infect? Does it need an open wound or can it colonize healthy tissue?
So that's a really good question. So far from our research, it needs a wound. So if the spores are just flying around and land on the bark, they will germinate, but they don't necessarily cause, they don't really permeate or cause infection. We are testing buds right now, though. We don't know if that's true for buds. Their morphology, physiology is a little different. But as far as like, actively growing wood, it doesn't seem to be able to penetrate. But if you wound it, they have no problem. They're actually very fast at causing colonization.
A couple more comments here on chat. This is a question about how old are the market vines and how often are the trunks renewed?
Oh, that's going to vary from grower to grower. I mean, I think for our survey, we had vines that were anywhere from 4 years old all the way up to 15 years old, I'd say. Maybe some that were older. And it's going to vary from grower to grower. We did ask them to provide some of that information, but I would doubt there's any correlation because we've surveyed so many places.
And then Craig is commenting that there's some growers, at least here in Wisconsin, that they are experimenting with cane pruning. So that might be one way of avoiding getting so many cuts and points of entry. John asked the time of treatment after pruning.
So for our experiment, we did it same day. So literally as soon as we were done pruning, we went back and applied all our treatments. So within an hour max.
Well, if anybody else has more questions, now's the time to. Oh, there we go. In New Hampshire, we use mostly cane pruning and have found that Marquette really thrives with frequent trunk renewal versus leaving older wood trunks. Good to know. I don't think that we see a lot of cane pruning here in the Midwest.
It's definitely new. I only have a couple of growers who are doing it, and I'm helping them quite a bit. We are doing cane trial pruning trials on campus now, so we'll have a little bit more demo. But I definitely think there's some hesitation. It's an unknown, so it's new for us.
Something new to learn, right? Are you going to sample more cultivars?
So if we can, we're trying to only sample ones that are symptomatic or at least have some symptomatic ones nearby. We do sample non-symptomatic ones, of course, to see if it's something that's just hanging out or chilling in the wood. But if a grower has them and they're having symptoms, then we will sample. So that doesn't mean that there's ones that are more or less resistant. We are doing some screening as part of an experimental process, but it definitely just seems that Marquette's the loser in this situation.
Do you know if any of the breeding programs are screening or are aware of this?
Maybe. This is pretty new stuff. This is the second time I'm talking about it in public, so I guess now it's going to be online forever. But
Yeah, everybody's going to see it now.
it's just sometimes when you find something new, look, there's a lot of questions, right? Something new. So we want to make sure we have all of our ducks in a row before we freak people out, before we do any trials with fungicides or different training processes. So that's what we've been doing.
Perfect. Well, it doesn't seem like we have any more questions. I just want to thank Suzanne for an excellent talk. A lot of great new information. We're hoping to maybe get you back with some of the new results that you're going to be collecting this season, and maybe some recommendations about how to treat this vine decline. So thank you so much. I also want to put a plug in there for our next webinars. If you're interested in some of the more webinars we're going to have, we are going to have another webinar. This is, if you want to just scan this QR code, this is the main page for the upcoming webinar. So we're going to have a webinar on H2A. We're going to have another webinar on insuring grapes and apples in Wisconsin. And our last webinar for this spring season on advancing cold climate grapes. And it's an update from the breeding program for grapes at the University of Minnesota. So feel free to just scan that QR code, and you can find there the links to register. And so we're going to leave it at that. And with that, just again, thank you so much, Suzanne. This is super informative and great research. And we're looking forward to hear more about your project.
Thank you.